Let us now discuss the different models of enzyme action. But before that, let us define first an enzyme-substrate complex. Enzyme-substrate complex refers to the intermediate reaction species that is formed when a substrate binds to the active site of an enzyme. So what is the active site of an enzyme? The active site is the relatively small part of an enzyme structure that is actually involved in catalysis. The active site in an enzyme is a three-dimensional entity formed by groups that come from different parts of the protein chains. These groups are brought together by the folding and bending of protein. They usually form a crevice-like shape. Let us now identify the 2 points of attachment in an enzyme. The substrate attaches to the active site of the enzyme. This is also known as the orthosteric site. Another one is the allosteric site. This is where another substance at a point other than the active site attaches. For this instance, we have inhibitors. Later on this presentation, we will discuss the different types of inhibition. Let us now discuss the two models of enzyme action, the lock and key model as well as the induced fit model. In the lock and key model, the active site in the enzyme has a fixed, rigid geometrical conformation. Only substrate with a complementary geometry can be accommodated at such site, much as lock accepts only certain keys. While in the induced fit model, it only allows for small changes in the shape or geometry of the active site of an enzyme in order to accommodate a substrate. The induced fit is a result of the enzyme's flexibility. It also adapts to accept the incoming substrate. The lock and key model explains the actions of numerous enzymes. However, it is too restrictive for the action of many other enzymes. The induced fit model somewhat contradicts the lock and key model. There are some experimental evidences that shows that enzymes have flexibility in their shapes. Let us relate these models to a real-life situation. I'm sure majority of you have experience playing with this type of toy. Let's just say that the base of this toy would be your enzymes, while the building blocks would be the substrate. The building block would only fit on the base that has the same conformational shape. This is one good example of the lock and key model. Let us now discuss enzyme specificity. This refers to the extent to which an enzyme's activity is restricted to a specific substrate, a specific group of substrate, a specific type of chemical bond, or a specific type of chemical reaction. Take note that the degree of specificity is determined by the active site. There are four main types of enzyme specificity. We have absolute specificity, stereochemical specificity, group specificity, and linkage specificity. In absolute specificity, an enzyme only catalyzes one reaction. This is the most restrictive of all specificities. One good example of an enzyme with absolute specificity is the enzyme catalase. Next is stereochemical specificity, wherein an enzyme acts only on a particular isomer. Another type is group specificity. It is the type where an enzyme acts only on molecules that have a specific functional group, such as hydroxyl, amino, or phosphate groups. One good example of a group-specific enzyme is carboxypeptidase. The last type is linkage specificity, wherein enzymes act on the particular type of bond, irrespective to the rest of the molecular structure. One good example is phosphatases hydrolyzed phosphate, wherein ester bonds in all types of phosphate esters are being acted upon too. As we have identified earlier the two main sites of attachment in an enzyme, let us now discuss enzyme inhibition. An enzyme inhibitor is a substance that slows or stops the normal catalytic function of an enzyme by binding to it. As a review, the active site wherein the substrate attaches to is known as the orthosteric site, while the other site is known as the allosteric site. The first type is reversible competitive inhibition. A competitive enzyme inhibitor is a molecule that sufficiently resembles an enzyme substrate in shape and charge distribution that it can compete with the substrate for occupancy of the enzyme's active site. When a competitor inhibitor binds to the enzyme's active site, the inhibitor remains unchanged. There would be no reaction occurring, but its physical presence at the site prevents the normal substrate molecule from occupying the site. The result of this is a decrease in the enzyme activity. The next type is reversible non-competitive inhibition. A non-competitive enzyme inhibitor is a molecule that decreases enzyme activity by binding to a site on an enzyme other than the active site. The substrate can still occupy the active site, but the presence of the inhibitor causes a change in the structure of the enzyme, sufficient to prevent the catalytic groups at the active site from properly effecting their catalyzing action. As you can see in the illustration on this side, the first one is an enzyme substrate complex in the absence of an inhibitor. The normal substrate can attach to the active or orthosteric site. The second image is a competitive type of inhibition. A competitive inhibitor binds to the orthosteric site, preventing the substrate to bind to the active site. Lastly, the last image would be the example of non-competitive inhibition. The non-competitive inhibitor attaches to the allosteric site even though the substrate can still attach to the active site. There is a conformational change in the shape of the enzyme affecting its activity. Correlate natin ang type ng inhibition na to sa isang relasyon. For the competitive type of inhibition, kunwari, meron kang boyfriend o girlfriend, harap-harapan na aagawin sa'yo ang jowa mo para makapag-attach sa kanya. While, sa non-competitive inhibition, hindi mo alam na meron na palang kumakabit sa jowa mo. The last type of inhibition is irreversible inhibition. An irreversible enzyme inhibitor is a molecule that inactivates enzyme by forming a strong covalent bond to an amino acid side chain group at the site of enzyme's active site. In general, such inhibitors do not have structures similar to the enzyme's normal substrate. The inhibitor-active site bond is sufficiently strong that additional of excess substrate does not reverse the inhibition process. Thus, the enzyme is permanently deactivated. This is the summary of enzyme inhibition.